Bacterial gram staining, microbiology laboratory skills. Introduction. Unlike simple stain, differential stain react differently with different kinds of bacteria and thus can be used to distinguish them. The differential stains most frequently used for bacteria are gram stain. The gram stain was developed in 1884, the Danish bacteriologist Hans Christian Gram. It is one of the most useful staining procedures because it classifies bacteria into two large groups, gram-positive and gram-negative. Gram-positive bacteria that take up and retain the crystal violet iodine complex, D, and gram-negative bacteria that take up the complex, but lose it on decolorization. The different reactivities are based on the different chemical makeup of the gram-positive and gram-negative organisms. Gram staining is often one of the first steps in identifying a bacterium. The gram reaction of a bacterium can provide valuable information for the treatment of disease. Gram-positive bacteria tend to be killed easily by penicillins and cephalosporins. Gram-negative bacteria are generally more resistant because the antibiotics cannot penetrate the lipopolysaccharide layer. Some resistance to these antibiotics among both gram-positive and gram-negative bacteria is due to bacterial inactivation of the antibiotics. In addition, a particular disinfectant may work against a gram-positive bacterium, but not one that is gram-negative. Gram-positive bacteria have certain structures not found in gram-negative bacteria, and vice versa. Bacterial cells are rich in nucleic acid, bearing negative charges as phosphate groups. These combine with the positively charged basic dyes. Acidic dyes do not stain bacterial cells and hence can be used to stain background material a contrasting color. The gram method is one of the most important staining techniques in medical microbiology. But gram staining results are not universally applicable, because some bacterial cells stain poorly or not at all. The gram reaction is most consistent when it is used on young, growing bacteria. Stains combine chemically with the bacterial protoplasm. If the cell is not already dead, the staining process itself would kill it. The process is thus a drastic one and may produce artifacts. The basic dyes stain bacterial cells uniformly unless the cytoplasmic RNA is destroyed first. Special staining techniques can be used, however, to differentiate flagella, capsules, cell walls, cell membranes, granules, nucleoids, and spores. In this video demonstration, we'll see about a differential bacterial staining technique, gram staining. This protocol is used to differentiate and classify the bacterial organism in two large groups, gram-positive and gram-negative bacteria. Material. The commonly used stains are salts. Basic stains consist of a colored cation with a colorless anion, example, methylene blue chloride. Acidic stains are the reverse, example, sodium eosinate. Crystal violet is a primary stain. Carbol-gentian violet gives a deep violet color to both the bacteria. Iodine is a mordant, Lugol. It fixes the crystal violet only to gram-positive bacteria because they have thick peptidoglycan layer. Methanol is a decolorizer, alcohol 96%. It decolorizes the primary stain easily from the gram-negative bacteria because the stain was not fixed with the gram-negative bacteria due to thin peptidoglycan layer, but the gram-positive bacteria retains the crystal violet. Carbol-fuchsin/safranine is a counterstain, fuchsin. It gives pink color to gram-negative bacteria. Procedure. Place the glass on the staining rack. Flood the slides with crystal violet or carbol-gentian violet. Be sure to fill the entire smear with the dye. Because the purple stain imparts its color to all cells, it is referred to as a primary stain. Let the stain on for 30 seconds. After the 30 seconds with the crystal violet, then dump the crystal violet in the sink and rinse it off until the crystal violet comes off. Don't over-rinse, and you can shake off the excess water. Now add the iodine or Lugol. This fixes the crystal violet in the cells. Let stain on for 60 seconds. So then after the 60 seconds with iodine, then rinse off the iodine. Shake off the excess water. And now we're going to decolorize using alcohol 95%. Decolorization is the most critical step. Now cover slide with safranine or fuchsin as counterstain. And let the stain on for 30 seconds. It's really important that you shake off the last water before this step so it doesn't get diluted. After 30 seconds, rinse off the safranine. Shake off the excess water. Then you can wipe the back of the slide on blotting paper. After the slide is completely air-dried, the smear will look like a thin colored haze. And your smear ready to observe under the microscope. How the gram stain works. The gram stain uses 4 stains/reagents. The results of the gram stain reflect differences in cell wall composition. Baraketnian gomungnya, per Urutanbaris D table. 1. Prepare heat-fixed film of bacteria on a glass side. 2. The primary stain enters the peptidoglycan of all bacteria, giving them a purple color. 3. The mordant attaches to the crystal violet to form larger complexes that prevent crystal violet from leaving thick peptidoglycan layers in gram-positive cell walls. 4. A decolorizer will remove the crystal violet/iodine complexes from the thin peptidoglycan layers of gram-negative cell walls, but not the thick peptidoglycan layers of gram-positive cell walls. After the decolorization step, gram-positive cells will still hold on to the purple crystal violet color, but gram-negative cells will lose the purple color and be transparent. 5. Safranine is a reddish-pink stain that will stain transparent cells, gram-negative cells, pink. After this step, gram-positive cells will still be purple and gram-negative cells will be pink. The cell wall confers rigidity upon the bacterial cell. The cell wall is a multilayered structure outside the cytoplasmic membrane. It is porous and permeable to substances of low molecular weight. The cell walls of gram-positive and gram-negative bacteria have important structural and chemical differences. The peptidoglycan layer is common to both gram-positive and gram-negative bacteria, but is much thicker in the gram-positive bacteria. Gram-positive bacteria don't have outer membrane, whilst gram-negative bacteria have an outer membrane. In contrast, the gram-negative organisms have a complex outer membrane composed of lipopolysaccharide, LPS, lipoprotein, and phospholipid. LPS released as endotoxins when gram-negative cells die. These form porins, through which hydrophilic molecules are transported in and out of the organism. The O antigen of the LPS and the lipid A component are also embedded in the outer membrane. Lying between the outer membrane and the cytoplasmic membrane of gram-negative bacteria is the periplasmic space. It is in this space that some bacterial species produce enzymes that destroy drugs such as penicillins, e.g. beta-lactamases. The LPS of gram-negative bacteria, which is extremely toxic, has been called the endotoxin. Hence, by definition, endotoxins cannot be produced by gram-positive bacteria as they do not have LPS in their cell walls. LPS is bound to the cell surface and is only released when it is lysed. It is responsible for many of the features of disease, such as fever and shock. Gram-positive bacteria cell walls additionally having teichoic acids and phosphate. The teichoic acids are of two types, one is lipoteichoic acid and the other is the teichoic wall acid. The microscopic examination. Bacterial identification initially entails examination of microscopic morphology and staining characteristics. What is the gram, positive or negative, of the cells? What is the cell shape of the cells? What is the cell arrangement? Then we categorize as any bacteria that may be present by color and shape during the microscopic evaluation. Color: typically, bacteria that are gram-positive appear purple, and bacteria that are gram-negative appear pink to red. Shape: the most common shapes include round, cocci, or rod-shaped bacilli. Arrangement: observing the groupings of the bacteria on the slide, examples include cocci that are present singly, in pairs, in groups of 4, in clusters or in chains. Bacilli that are thick, thin, short, long, or have enlarged spores on one end. If bacteria are present within white blood cells. Common mistakes. 1. Smear preparation. Beginning students tend to make the smears too thick by putting too much inoculum on the slide. This will make it harder to properly decolorize the bacteria. Always put saline or water on the slide before or after adding bacteria when using a culture from solid medium, slant or plate. The amount of inoculum from a solid media should be a pinpoint amount. You should just barely touch the organisms you want to stain with your loupe. Note: you do not need to put saline or water on the slide when making smears from a liquid media. 2. Allow the smear to air dry. This means the slide must sit until it has no longer appears moist. Alternatively, you may use a slide warmer to dry your smear. The slide warmer will dry the slide faster. When using the slide warmer, do not take your eyes off the smear. As soon as the slide is dry, remove it from the slide warmer. If too much heat is used, the cell wall of the organisms may be distorted or rupture and release the crystal violet dye when it should retain it. Extreme overheating may cause the entire cell to lyse, and you will see nothing under the microscope. 3. Heat fixing will adhere the bacteria to the slide and kill the bacteria. If you do not have any cells on the slide, likely the heat fixing step was skipped or not enough to fix the cells to the slide. If you are staining a mixture of gram-negative and gram-positive bacteria and you do not see cells, no cells are visible. It is likely because the smear was not properly heat fixed and the bacteria washed off the slide during staining. 4. Freshness and concentration of reagents. Reagents must be fresh, and concentrations must be accurate. 5. Failure to drain slides before adding next reagent. After you wash a slide with water and before you add the next reagent, you must be sure to tilt the slide to allow excess water to drain off it. If you leave a bubble of water over the bacteria, the reagent you add may never reach the bacteria or it will be greatly diluted by the water present on the slide. 6. Age of the bacterial culture. More than 24 hours old gram-positive organisms lose their ability to retain the violet dye as they move out of the exponential growth phase. The age of the culture. 7. Timing of the decolorizing step. This is the most critical stage of the procedure. If the decolorizer is left on too long, gram-positive organisms will appear gram-negative. If the decolorizer is not left on long enough, gram-negative organisms will appear gram-positive. The thickness of the smear will dictate how long you will need to decolorize. It is impossible to give an exact time. You must decolorize one smear at a time and watch it closely. When the color begins to lift off the smear, you should wash off the decolorizer. If you are staining a mixture of gram-negative and gram-positive bacteria and you see all cells purple, it indicates that the decolorization step was too short. The next time you do a gram stain, increase the time for decolorization. If you are staining a mixture of gram-negative and gram-positive bacteria and you see all cells pink, it indicates that the decolorization step was too long. The next time you do a gram stain, decrease the time for decolorization.